Good afternoon and welcome to NASA's Wallops Flight Facility. We're here today for the What's On Board science briefing. This is a live chance to talk to researchers who have science going up to the International Space Station on Northrop Grumman CRS-10. The Cygnus spacecraft, which you see on your screen there, is on top of the Antares rocket. It's filled with about 7,500 pounds of science, research, and cargo for the International Space Station. And today you're going to get a chance to talk to those researchers. You're going to get to see their science that they have going up. And first we're going to introduce you to Dr. Tara Rutley and Dr. Liz Warren. Dr. Tara Rutley is Associate Chief Scientist for Microgravity Research in NASA's Office of the Chief Scientist, and Dr. Liz Warren is the Associate Program Scientist for the ISS National Lab.
Okay, thanks everybody. It's really great to be here. I'm Tara Rutley. I am looking forward to listening to the researchers for the next hour or so talking about how the space station environment enables them to perform their studies and their tests in a way that's not always so intuitive as compared to how they're trained to think on Earth, where there's always a gravity vector. So we'll share with you some examples today of how the research that's headed up on NG-10 contribute to what we need to do now to be able to enable human exploration beyond the Earth orbit. And you'll also hear from some researchers who find the real need for use of the microgravity environment to advance their studies here on Earth. First, I want to share with you one of the experiments that we are launching on NG-10. You'll hear from these researchers shortly. It's enabling human exploration, but it's the first ever Refabricator being sent to the space station. So it's the first time we will ever try to recycle plastic parts and print from those, 3D print from those parts as well. So we're thinking again about enabling human exploration, the things that we need to do now while we have access to this low Earth orbit environment that's going to get us further. The second investigation I want to share with you today, on behalf of some of the researchers who won't be here from the University of Pennsylvania, is an investigation called MVP Cells 5. It's a cement formation process investigation. And can we put the first image up on top, please? This team is actually going to look at how cement forms in a microgravity environment. In particular, on this particular mission, they're going to look at how cement formation happens in different gravity vectors using a centrifuge we have on orbit. So this team actually sent up some samples to the space station early this summer and got some samples back after they grew these microstructures through their formation in space. So what you see now is, I think it's called Portlandite. It's a type of material that's used to form concrete through cement formation processes. This is an image taken that the investigators sent me on Earth. And you can kind of take this mental snapshot right now, because I'm going to show you the next slide, and you can think about what's different in this particular slide. So does anything stand out to you? Probably that big crystal in the middle, right? So this is an example that the investigator shared with me to share with you of some of the samples, microstructure that they got from their flights earlier to space station. Now that's a crystal that they had not really expected to see, and that's a result of the lack of sedimentation and convection that they get when you're in the microgravity environment. You don't have that force of gravity interfering with crystal growth. So these scientists are looking at the microstructure of concrete. Now think about that. This particular investigation plus the Refabricator are ways that we are using the space station to develop like the infrastructure. Think about how we would create things on the moon or on Mars as we go beyond low Earth orbit. And also interestingly, this afternoon you'll hear from a group of students who are also doing their own concrete investigation experiment, which I'm not going to give away the details. I'm going to save the fun for then. But again, it's part of advancing our human exploration beyond low Earth orbit. We have another investigation that comes from our Canadian partners. They are also not here today, but I wanted to share with you, again, looking at how humans can live in space longer, they are performing an investigation called VECTION. And VECTION is where humans perceive motion based solely on visual cues. So they're looking at what happens to astronauts in the microgravity environment when you don't have the visual cues that are as constant as they are on Earth, and your vestibular system is challenged, and your proprioception system is challenged. All these sensory inputs are challenged. What does this mean about how they perceive their own self-motion, and also how they judge distance in space, too? Because these are critical things that we need to study to keep our crew healthy. So those are some quick examples of some of the human exploration investigations that are going to be enabled by NG-10 launch. A little bit closer to home, you'll hear from Liz Warren from the National Laboratory efforts on how these scientists that you'll hear from this afternoon are doing things to advance their own research here on Earth that could benefit you and I as well. But before I turn it over to Liz, if you want to know anything more about the space station science that's happening, you can always go to www.nasa.gov/ISS-science. Thanks.
Thank you, Tara. So as a national lab, the space station is open to users in industry, academia, and other government agencies, and enables scientists to look at their research through a new lens. It enables, it's almost like additional discoveries or additional benefits that are for the benefit of all of us here on Earth. Three unique selling propositions or USPs of the space station, of course, include microgravity. There's also an exposure facility outside the space station for testing materials in a very extreme environment. And then, as a platform about 250 miles above the surface of the Earth, it provides an opportunity for Earth observation and remote sensing. The ISS National Lab investigations on board Northrop Grumman 10 span an array of research disciplines from life and physical sciences, as I mentioned, Earth observation and remote sensing, and as well, student payloads. Actually, let's roll the video and you'll see some of these examples.
5, 4, 3, 2, 1, and we have ignition.
Northrop Grumman's 10th resupply mission to the International Space Station carries an exciting array of research that has the capacity to benefit life on Earth. A lot of those payloads are sponsored by the U.S. National Laboratory. Let's get an idea about some of the major payloads that are associated with this mission.
The Cygnus continues to close in, robotic arm capturing the Cygnus.
The Michael J. Fox Foundation is continuing their research and development pursuits on the space station as they seek to grow crystals of and better understand the LRRK2 protein in microgravity, which is closely associated with the development of Parkinson's disease. Multiple payloads on this mission will leverage the extreme environment of space by being placed in the Missy Flight Facility outside of the space station. Materials that are placed in the extreme environment of space can experience vast temperature changes and greater levels of radiation, both of which can enhance knowledge on the performance of a product. Semska, a startup company in Houston, seeks to leverage the microgravity environment to improve the design and manufacturing of cost-effective membranes for separating carbon dioxide gas molecules from the air and other gases. Such membranes could be used in gas separation technologies in fossil fuel power plants towards reducing greenhouse gas emissions. This launch represents the diverse research potential aboard the station, from life and physical sciences, manufacturing, and student investigations. To learn more about all the investigations on this mission, visit issnationallab.org. Best of luck to our partners at Northrop Grumman, and go Cygnus.
And so you'll hear shortly from some of these researchers. They are very excited. They've worked very hard to get their research ready to go. We're all very excited to see their science on board space station. And when you're out there watching the launch and you're saying, go Antares, go Cygnus, add a go science in there too. And if you have any questions,
up to questions for those in the room. We also have the phone bridge open, and if you're following us online or on NASA TV, you can ask questions using the hashtag AskNASA. All right, and so we'll open up to questions in the room. Ken Kramer?
Hi, Ken Kramer, Space Up Close for you guys. Are you finding more and more people and universities and companies interested in working with the National Lab?
Well, absolutely. Now, of course, there's a multitude of mechanisms to get research on board the space station. There's answering calls from NASA solicitations. There are solicitations from the National Lab, and then there's unsolicited mechanisms as well. And we are seeing more and more users. There's small companies, big companies, academics as well. And all of these folks are interested in really exploiting those very unique research opportunities on board the space station.
All right, and we'll now go on to our next two presenters. Thank you very much for being with us. So now I'd like to introduce you to Diane Risden and to Dr. Allison Porter. As they're making their way up, Diane is from In-Space Manufacturing on the Refabricator project, and she's the lead at Marshall Space Flight Center. And then we have Dr. Allison Porter, the flight mission manager at Tethers Unlimited.
Good afternoon. We're very excited. We're thrilled exactly to be here, and we're looking forward to launch on Thursday. Probably the first question that you all are having is, what exactly is the Refabricator? The Refabricator is a combination 3D printer and recycler, and here it comes. And it was designed and developed through a small business contract with a company, fantastic company, Tethers Unlimited. On the screen coming up next is the In-Space Manufacturing logo for Marshall Space Flight Center. It's our motto is make it, don't take it. In-Space Manufacturing is, we focus on rapidly evolving disruptive technologies that'll take us from the ISS to the lunar mission to deep space exploration. And we're leveraging small businesses such as Tethers Unlimited through the Small Business Innovative Research called SBIR program, which you may have heard of. We have a project manager named Nikki Werkheiser, who's I call her the visionary. She's developed a roadmap that shows in detail all of the new technologies that we're going to need to get us from ISS to lunar to beyond lunar. And so we focus on all of those steps. Each one is a stepping stone to the next technology that we're going to use. So first off, when we're going to go to space, the main thing is that we have to have spares. For every consumable, there has to be a spare. So if you can imagine the amount of spares that we need, it's a very tremendous mass that has to be launched to space. And not to mention the cost involved with that, because 1 pound of mass costs NASA thousands of dollars to upload to space. So the first step in our roadmap was, how can we help out with this? What can we do? 3D printing came along, and we decided, let's do a technology demonstration to see how microgravity affects 3D printing, because it had never been done before. So through a small business contract, 2014, we set up the very first 3D printer in space. It was installed into the microgravity science glove box, where it printed 55 parts. And those 55 parts were returned to Earth, where they were tested and analyzed just to see what the effects, if any, of microgravity would have on 3D printing. And when all the results came in, we found out that there were no engineering significant microgravity effects. So now we have our 3D printer. We know that it works in space. The next thing is, where do we get the filament? How will we have the filament? Do we have to upload that? Well, we're trying to avoid large masses being uploaded, so we've got to find a sustainable source for filament. So on the ISS, we know that there's multitudes and multitudes of plastic baggies. The crew complains a lot of them, what do we do with all these baggies? They also have packaging, plastic packaging. They have used food, plastic containers, plastic medical devices. So periodically they round up all these trash, as they call them, and burn them in space. So they all have in common that a lot of them are made out of plastic, so we're thinking, well, there's our resource. If we can recycle these, then we're on our way to getting our filament. So also in 2014, Tethers Unlimited was awarded a small business contract to design the very first recycler, and we decided to make it a technology demonstration. But Tethers, being the forward-thinking, innovative company that they are, took it one step further. And they proposed on their own, hey, we're going to go ahead and build you a recycler, but can we go ahead and build a 3D printer as well to include in one unit, closed loop, so that we can control our processes, and more importantly, it cuts immensely on crew time because you have recycling and printing all on the same unit. You don't have to have astronauts moving from one unit to another. So here's this is a mock-up of the hardware just so you can get a general size. It's about the size of a dorm refrigerator, and it fits into double locker on the ISS Express rack, I'm sorry. So I'm going to turn it over to Dr. Allison Porter now, and she'll tell you more about the Refabricator.
Thank you, Diane. So as Diane said, I'm Dr. Allison Porter. I'm the Flight Missions Manager at Tethers Unlimited. And Tethers, we developed, designed, and tested the Refabricator. So this is the mock-up, as Diane mentioned. So basically, what we're taking is we're melting down polymers and making it into 3D printer filament. So here's an example of a spool of filament that we recycled at our lab. This is very similar to the filament that we will be generating on the space station. Once the Refabricator recycles and makes new filament, we are able to print new parts. The parts that we are printing are going to be primarily these tensile specimens that I'm holding here. This will allow us to have samples of not only the 3D printer filament that we're recycling, but also of printed parts generated from that filament, and that will allow us to test these specimens on the ground and see the effects of recycling this plastic, not just once, but multiple times. So we'll be recycling these plastics up to 7 times on the space station to see how strong is the plastic after being recycled and processed over and over. So it's not just about breaking down and using the things that we have on station available to us, but being able to do it over and over again to create a more sustainable in-space economy. So Refabricator is really just, as Diane mentioned, is a tech demonstration. So the focus is on the science, on the recycling technology that Tethers developed, but it's just a stepping stone in really creating the full manufacturing capability of polymers in space. So the recycling. So even though this is a focus on the recycling technology, because we integrated a 3D printer, that allows us to look at the closed loop cycle of the plastic processing, and then will also enable us to take the lessons learned from Refabricator and to develop future polymer technologies. So one of the payloads that we are working on or one of the technologies we're working on is CRISP. So this is the generation of launch packaging material or other packaging material that can be made with recyclable materials. So this is an example of something that could be recycled and then made into new parts after like hardware or other tools have been safely delivered in the packaging. It can be broken down and reused. Another technology that we're working on is called Erasmus, which is our ability to sterilize and print food-grade and medical-grade items out of polymers. So both CRISP and Erasmus take everything that we're learning from Refabricator, from the recycling, from doing very autonomous operation in a single closed box and taking it to the next level in terms of how can we better utilize polymers in space. And this also folds into a lot of the work that Tethers is interested in terms of developing that sustainable in-space economy where we're building what we need, we're manufacturing what we need, and assembling what we need in space rather than just delivering all of that directly. So with that, I'll hand it back to Diane.
And we did have a video. Can we show the video of it printing? We have a video of the actual Refabricator printing. I want to point out that it's a very new novel process. On Earth, a lot of the plastic goes through a grinding, and you lose some of the material properties while that happens. So they melt the plastic. It's a novel process, brand new cutting-edge technology. So anyway, in closure, I just wanted to say that we're The ISS is an amazing test bed for testing all of our technology demonstrations. We've done the 3D printer, first recycler. There's more coming from in-space manufacturing as we move forward. And we were so grateful to work with this company. They're a small company, but they're filled to the brim with brilliant minds, let me tell you. They're super people, and we've benefited immensely from working with them, their expertise and knowledge. They took an idea that we started out just with a recycler. They added to it, made it a closed loop, turned that novel idea into something that's going to fly. And we're so excited this Thursday that our payload is going up. So thank you for hearing our presentation.
And now we do have some time for questions, so we can ask questions for here in the room. And remember, for those following on social media, you can ask questions using the hashtag AskNASA. So we'll go to here in the room first.
Hi, my name is Haley Harding. I'm a reporter with the Salisbury Daily Times. How much did this cost to produce, and how much do you expect it will save you guys in the long run?
Well, we can't really come up with a total price tag. We'll say it's priceless to us. It's gone through several iterations. Tether started out with what they call the positrusion, which is the recycler portion, and it's carried forward. So it's hard to put like a final price tag on it. We're going to save immensely, efficiency, schedule. So we just say it's priceless. That's pretty much all we say. But it's helping immensely with reducing mass cost and mass upload and cost to get everything up into space.
So we're going to do another question here in the room, and then we'll take one from the phone.
Okay. Alex Mankiewicz of the Zog 43 newsletter. Can you speak a little bit more about the ancillary benefits from the refabrication? For instance, does it have inherent sterilization ability? Does it have inherent deodorizing ability?
So the sterilization of recycling and 3D printing is something currently under investigation through our Erasmus grant that we're under, also with the in-space manufacturing program. The inherent sterilization of Refabricator specifically has not been studied, but Erasmus will take a lot of the technology in the very similar way of recycling and printing to investigate that specifically. So hopefully we have results on that sterilization properties, hopefully in the near future.
And now we'll go to the phone bridge.
Yes, there's a question in the queue from Jeff Faust with Space News. Your line is now open.
Hi, I actually had a question for the first round of speakers, not this current experiment. Sorry.
There is another question in the queue from Sarah Lewitt with Space.com. Your line is now open.
Hi, I was curious whether any of the plastics already up there are harder to recycle than others, or if they're all pretty much the same.
What was the question again?
And could you repeat the question?
Sure. If any of the plastics already up there are harder to recycle than others.
So in terms of difficulty of recycling plastics, a lot of it comes down to what the melting temperature of the plastic is. So the plastic that we're doing for Refabricator is on pretty much the upper end of the polymers that are available on the space station. So really the idea would be to start with the hardest thing and then know that we can tune our temperature profiles to recycle other plastics. So we will be able to cover, especially in the future generations when we're looking at using multiple materials with a single technology, that we will be able to process most of the plastics that are up on the space station that are capable of being recycled.
And we're going to go back to that phone question on the phone bridge if we can.
Yes, Jeff, your line is open.
Hi. Yeah, my question is actually for Dr. Rutley from the first round of discussion about the effect the reduced crew complement on the space station has had on ISS utilization in terms of number of hours available for experimentation and whether that's affected plans for experiments flying on this mission or future resupply missions. Thanks.
No, okay, thanks. Good question. No, we are still meeting record crew time hours. We haven't needed to readjust any of the research planning truly associated with the decrease in crew size. We thought we might have had an issue a few months ago, but as the planning process has worked out, we're meeting the needs of all the researchers and meeting the research plans. So there have been no major impacts. The crew's staying busier than ever. Again, record number of hours. Even with the Soyuz anomaly, we have not had any change to the investigations that are on this particular launch, and I just learned that we haven't had any changes to the investigation on the SpaceX 16 launch as well.
Okay, and now we have a question from social media.
Twitter user Jean would like to know, if the Refabricator is a success on orbit, what do you see for possible Earthbound applications?
So since we do use a lot of polymers on Earth, we have boundless amounts of water bottles, plastic bags, things like that. Refabricator does have applicability to Earth applications. A lot of what is specific to this unit here, and the one going on station, is very particular about being installed into the express rack, but the basic technology, the idea and the technology development behind the recycler itself and the 3D printing can be very easily adapted to an Earth version of Refabricator that's more tuned for everyday use, for commercial or industry applications. And then I guess another good thing to point out is that our recycling technology actually produces higher quality filament than a lot of the commercially available filament. So even as just a standalone recycler, Refabricator offers an alternative means of generating 3D printer filament to produce higher quality parts on the ground.
And thank you very much for being with us.
Thank you.
And we'll now introduce our next speaker. It's Dr. Sivan Mullaney, founder and president at MicroGRX.
MicroGRX was located in Orlando, Florida. It's founded to develop human cell-based lab-on-a-chip models for use in space. And so right now on the Cygnus, we're culturing and growing human primary muscle cells in these small chips. And we have 2 chips in our payload. There are 4 chambers on each, so we have 8 separate experiments. And they're isolated from muscle biopsy, the quadricep muscle of 8 donors. And so the first slide, please. And we've worked with Florida Hospital to collect these samples from individuals that are phenotyped both on age and physical activity. So what's flying on a Cygnus is from donors that are young athletic donors, because we feel we wanted all of the 8 experiments to be the same experiments to get our statistics, and feel that that microgravity would have the most change on these the young athletic donor cells. And so these cells, and you can see a picture of the cells, the precursor cells are grown in a certain media, and then they differentiate to form the fibers that we know muscle fibers. And you can see those in the red outline. This is what they look like on Earth, and we're going to be excited to see what they're looking like on their voyage to the ISS. The blue circles are the nuclei with the genetic material. So we're going to send these. They'll have about 7 days in microgravity, freeze them, preserve them, bring them back, and we'll do second-generation sequencing, gene expression studies to look at the changes induced by microgravity. So this is a cell biology experiment looking at microgravity effects on the muscle cell biology in this experiment. And the idea really being that we know that microgravity has detrimental effects on skeletal muscle from the astronaut studies. And so on a cellular level, what can we see in these cellular changes in the cells, and that can be induced in a more accelerated time frame. So somewhat mimic the age-related process of muscle wasting. That's called sarcopenia. There's no really available therapeutics approved yet for that. So we're really looking at a disease model and what we can learn on the cellular level, and then continue to advance the science in future flights. So how do the cells get there? The really unique thing about our payload is that it is all contained in a 30 by 10 centimeter box. So it's a miniaturized lab. And there's really no astronaut intervention needed except to put it in the freezer at the end. And everything's contained in there, from the pumps that deliver the fluidics and the media, the nutrients. It's an incubator in there, so it has the CO2, the humidity required for cells to grow and survive. The platform is heated to 37 degrees, which is where our cells are the healthiest and the happiest. And here you see on the camera system we have installed in the box, the optics will actually move in the XYZ plane, so we'll have different field views from each of the chambers, and also in the Z range, we'll have different focal planes so that we can get the right focus and take as many images as we can store during the voyage to the ISS. And so this has been an international collaboration. We have seed funding from Space Florida through the Florida-Israel Innovation Program. So slide 2, please, is the team arriving at the Wallops facility. It's really the first time that we came together as a team to integrate the technology with Space Pharma, who is a company developing the miniaturized lab-on-a-chip devices and millifluidics, with the Florida team to integrate the science. And we're working out of the Wallops laboratory, which really was dusted off after a couple of years of no use, and will continue to get more use as a wet lab space. Slide 3 is a fun time lapse of us in the laboratory working with the cell culturing, tissue culture. We were soldering until hours before launch. We were using the microscope and really working for about 5 days around the clock to assemble the payload and get it ready for space. So this is a kind of refurbished lab, which will continue to get more use, I think, in the coming, as we have 24-hour turnovers. And so the next slide actually shows us inserting the cells into this miniaturized laboratory. And it was the last thing that we did. So you can see the injection there into the port. I don't think you really liked me standing over his shoulder saying, is that the right port? Because there's a number of ports there that go to the waste, go to injecting the media into the reservoirs. We have a preservation mix that the cells will get after their 7 or about 10 days after growth. That preserves it. We stick it into the polar freezer. It comes back on the Dragon SpaceX. And then we'll do our gene expression changes studies post-analysis. So we did have a control box going at the same time so that we injected the cells. We could look at them a couple hours later before we handed off to show that we had cells and they were growing fine. And so the next slide shows us going into the Merlin power locker. And the Merlin keeps the temperature at 4 degrees. And so the platform the cells are on are heated to 37. It's a lot easier to heat than use power to cool, because we have a lot of power that we need to operate the pumps and the fluidics. And so there it is. It's connected to the Space Tango power cube to give it power and connection in the locker. And then the whole system is at 4 degrees, but our cells are still at 37. It will stay there until we add the preservation. And then the only astronaut intervention is to take it from the Merlin and put it into polar, which is a minus 30 degree temperature, and we'll wait to come home on the Dragon and do our analysis. So this really sets the stage for tissue chip, organ-on-a-chip studies that are coming in the coming months on future flights, and looking at various disease models and microgravity-induced degenerative models. And so we've been funded for 2 more flights after this, and so our cell culturing device will become much more of a tissue chip device where we'll be looking at muscle contraction, so we'll have the ability to look at that as well as cytoskeletal changes, and much more looking at the changes in strength and muscle mass that will be much more physiologically relevant and we'll have 4 chips, so 16 samples, and really increase and expand the science. But this is a new capability on the ISS, and we're just really excited and excited for a great launch in the wee morning hours of Thursday. So thanks. And acknowledgement slide here. So the last slide is just showing our partners in this, Space Pharma of Israel, Florida Hospital, where we had the samples. Space Tango is our implementation partner and hosting us on the ISS. And then we had the funding from Florida-Israel Innovation Partnership from Space Florida and Organ-on-a-Chip Challenge from CASIS, which gave us the opportunity to fly the system.
And thank you very much. And as a reminder, you can ask questions online using the hashtag AskNASA. But first, we'll open it up to the questions for here in the room.
So I assume that you're using quadricep muscles because they reproduce rather quickly. How do you think muscles that reproduce slower would be affected in this study?
What I hope to see is that we have all the different cell types. So we've also have samples from old sedentary individuals, sarcopenic, which are the age-related muscle wasting. So we have all these samples, and wanted to send different sample types, but we're really limited on space, and so we're just sending the ones that are the young athletic. But with the gene changes, we hope we see this convergence or this degeneration that maybe mimics or looks more like what the old sarcopenic cells may look like. So we'll have those comparisons. In our future flights, we'll be sending different cell types from different individuals that are phenotyped from age and physical activity, and be able to have some comparisons on orbit as well as on ground.
And additional questions here in the room? And I do believe we have a question from social media.
Twitter user Jason would like to know, will the results of the experiments be available to the public?
They will, and in fact, the gene expression changes will most likely be put into Gene Lab, which is the NASA database, and as part of funding, it's deposited for public use.
Thank you very much for being with us today. All right, and our next speaker is Michelle Lucas, who is the founder and president of Higher Orbit.
Good afternoon. It is really great to be back here. We were here about a year ago. It was a little bit colder than it is hopefully going to be this week last year. I'm Michelle. I'm with Higher Orbits. We are a 501c3 nonprofit that uses spaceflight as a way to get students more engaged in science, technology, engineering, and math. While doing that, we also work on their teamwork, leadership, and communication skills, and I think you're going to see that is very apparent with the group that we have here today. We hold a program called Go for Launch, where for multiple days, students in grades 8 through 12 can compete to have their science ideas flown to the International Space Station. We pick winners from various events, and the overall series winner gets to have their experiment launched to space and carried out. They'll get the data back, analyze it, and work with scientists to make sure that it is really well analyzed and collated and all that. But I'm not going to tell you about the science, because the reality is I don't know much about this. This topic is something that is near and dear to these students' hearts. Every experiment we've had has been different. This is our 7th different experiment that has flown to space, and as such, we've had 7 different, very unique experiments, and there's no way I could know enough about all of them. So I'm going to turn it over to the experts. We have 3 of the team members here from the 5. We have Ryan, Spencer, and Tyrion, who are here. If you could show the picture real quick, we'll see the picture of all 5 team members. The other 2 will be here for a launch, but they were kind of busy with school today. But for now, I'm going to hand it over to these gentlemen.
Hi. We want to preface our conversation today with a big thank you to Michelle, Space Tango, Higher Orbits, and our families for all their support throughout this entire process. We couldn't have been here on this stage without their help. Now, with that being said, hi everyone, I'm Spencer.
I'm Tyrion.
And I'm Ryan. Along with our fellow teammates Abby Maltese and George Tang, we were the members of Team Waffle, the winners of the Cygnus Series Go for Launch competition.
So you guys are all probably wondering how we got to where we are right now. So this endeavor started in early February when we attended the Go for Launch event at the headquarters of Northrop Grumman Innovation Systems. Over the course of 2 days, students formed teams of 4 to 5, competed in a series of STEM-oriented challenges, such as the Adhesiveless Construction Challenge, listened to lectures given by former astronauts and many professionals, enjoyed snacks of all varieties, and polished up on their research and presentation skills.
All the students who participated in this event, while doing all of those many different tasks, also formulated, created, designed, elevator pitched, and presented an experiment with the main focus being the effects of microgravity. This was all to achieve the end goal of having that experiment put on the International Space Station.
Now for some information on our actual experiment. Our investigation concerns the effects of microgravity and low Earth orbit conditions on the basic principles of self-healing polymers and, well, specifically a self-healing concrete. These materials have the ability to mend themselves without external diagnosis or human intervention. The concrete in our experiment functions using spore-laden bacteria colonies and a chemical healing solution.
We believe that the microgravity will influence these bacteria in a way that could increase the efficiency of their healing process or calcification process. It could also affect the structure of the calcification produced. And we believe that this could all be done without any influence of an astronaut.
So we believe that there are several applications for these self-healing concretes in the future of aerospace and space exploration. Now, one of these applications is the creation of structures on other planets. The self-healing properties of these concretes make it such that less time is necessary in order to repair structures such as microfractures and other small damages.
Another application for self-healing materials as a whole would be for a material that could be used on vehicles that normally take a lot of small damages, microfractures, such as a replacement for the space shuttle.
We hope that our experiment is able to shed some light on the nuances of self-healing materials in microgravity. We hope that it expands the research of other self-healing materials in microgravity conditions, and it spurs the application, the discovery of the applications of self-healing materials in other fields as well.
We'd like to say one last thank you to Michelle, Higher Orbits, and everyone who's helped us throughout this process. We will now take questions.
And with that, we will start taking questions. We'll begin with any questions here in the room.
Tom Hench, Bayside Studio, do you see any possible correlation with the first presenters talking about the 3D printing and what you're doing?
As it would happen, we never planned any of this. Yeah, it just happened that way. There's also another experiment concerning concrete where they're seeing how concrete forms. That was completely unplanned too. It just sort of happened that way. Just pure coincidence.
Doug Money, Space IT Bridge. To follow up on that point you had, so your experiment is independent of what's happening at Penn State with a concrete fly. Do you plan to maybe do some mini workshop or brain share or something with them in the future?
So we've just learned about the Penn State science. We are in process of signing some MOUs with various universities to help these brilliant young minds further analyze the data, because while they've got a lot going on, having some scientists who've been working on it a little bit longer might be helpful. So yes, we are always open to collaborations, and we are collaborating for all of our previous experiments as well with universities and researchers. So yes, as they get busy with school, we're going to make them even busier with all the extra.
And any further questions in the room? All right, well, thank you very much for being with us. And thank you all for joining us for live coverage of NASA's What's on Board briefing, which covered just some of the science headed up with Antares. That launch is going on November 15th at 4:49 a.m., but set your alarms a little earlier because NASA television coverage will begin at 4:15 a.m. Thank you very much. Have a good day.